Hello, dear students. So, we'll be continuing our class on molecular endocrinology. So, the mechanism of action, we have taken into consideration the first two, that is the interaction with nuclear chromatin and membrane receptors. The third is stimulation of enzyme synthesis at the ribosome level. So, at the ribosome level means at the level of translation, and the modified ribosomes from growth hormone treated animal can be one example. The capacity to synthesize protein in presence of normal mRNA is also taken into consideration. It is simply the fact that when you stimulate enzyme synthesis at the level of translation, either the production, either there is increased production of new ribosomes or it creates a new population of more active selective ribosomes. So that is what is indicated in this. The fourth one is the direct activation at the enzyme level. So that is a bit difficult to demonstrate, and the treatment of intact animal or isolated tissue has to be taken into consideration to actually understand the direct activation at the enzyme level. So there are extremely rapid changes that happen in this process, and the cell membrane is usually required. So it initiates a hormonal event causing the activation of membrane receptor. So next is the cyclic AMP and hormone action. So cyclic AMP, as you can see, is the second messenger, and cyclic AMP is produced by inactive adenyl cyclase, by conversion of inactive adenyl cyclase to active adenyl cyclase. So where the mediator becomes the G protein, the receptor is linked to the G protein which is inactive, and when the receptor binds to the hormone, the G protein gets activated and it dissociates to its subunits. So the dissociated activated G protein which has a GTP bound will go and activate the adenyl cyclase so that ATP is converted to cyclic AMP. So the action of cyclic AMP activates cyclic AMP dependent protein kinase. So cyclic AMP dependent protein kinase has four subunits or four, it's a homodimer. Dimer means two subunits, and homodimer means two of the subunits are equivalent or they are the same. Okay, so you can see that there are two R's and two C's. R stands for regulatory subunit, C stands for catalytic subunit. So the regulatory subunits, in presence of cyclic AMP, the regulatory subunits are removed and the catalytic subunits are released, thereby becoming active. The catalytic subunit converts the protein substrate to a phosphorylated form of the protein, and the phosphorylated form of the protein mediates other intracellular effects. So phosphodiesterase is the protein or the enzyme which converts or inactivates the cyclic AMP to AMP, 5-AMP, so that the stimulation does not happen continuously. It's a form of regulation of cyclic AMP action. So hormonal stimulus, how do you stop the hormonal stimulus? That's why that's what I said right now. So the release of hormone from its receptor, which is an unbound receptor, and dephosphorylation of protein substrate by phosphatase, degradation of cyclic AMP into AMP. biphosphodiesterase. So protein kinase A becomes inactivated by the absence of cyclic AMP, and GTP is hydrolyzed to GDP. So as it goes into the GDP state, the alpha subunit and the beta gamma subunits of G protein will bind together, and as a result, it causes the inactivation of adenyl cyclase. So we'll be seeing this in detail as we go further. 6 and 7 both have been highlighted because both are very similar in its action. So role of polyphosphoinositol or phosphatidylinositol and diacylglycerol, and the role of calcium in hormone action. So in this process you can see this is the receptor. The receptor binds to the hormone. The hormone has a bound G protein to it, which gets activated, releases the beta gamma subunit, and the alpha subunit with the GTP bound binds to phospholipase C. So till the alpha subunit becomes activated and binds to a receptor or a integral membrane protein, till that it's the same as for cyclic AMP. But for cyclic AMP, the alpha subunit activates a adenyl cyclase. Here the alpha subunit activates the phospholipase C. So the phospholipase C then converts phosphatidylinositol from where? From the cell membrane. So phosphatidylinositol is present in the cell membrane, and the phospholipase C converts phosphatidylinositol to diacylglycerol and inositol triphosphate. So inositol triphosphate has a function of binding to the calcium channels of endoplasmic reticulum, thereby causing release of calcium ions. The diacylglycerol has the function of binding to protein kinase C, which also increases the calcium concentration inside the cytoplasm. So all of these processes, the presence of calcium, which activates the protein kinase C, and the activation by diacylglycerol. The activated protein kinase C further phosphorylates proteins, which causes the intracellular effects. So the calcium, this is the structure of the calcium phosphatidylinositol system, and phospholipase C acts at this particular position. Sorry for the disturbance. So this is the structure of calcium phosphatidylinositol system, and this is where the phospholipase C actually affects. So this portion is the diacylglycerol, and this becomes the inositol triphosphate. So the next one is the role of cyclic GMP in hormone action. So just like you have cyclic AMP, you also have cyclic GMP. So the cyclic GMP is very specific. That is very few systems use cyclic GMP in hormone action. So one of the biggest examples is the atrial natriuretic peptide hormone, ANP hormone, which is a cardiac hormone, and the main function is to lower blood pressure and control electrolyte homeostasis. So this activates PKG. So what you have to consider is cyclic AMP activates PKA, calcium activates PKC, cyclic GMP activates PKG. PK stands for phosphokinase. Sorry, protein kinase. So protein kinase A, protein kinase C, protein kinase G. So CGMP activates protein kinase G, which inhibits the action of tumor growth factor through the phosphorylation of SMAD3, preventing its heterodimerization of SMAD4. So don't get overwhelmed by that. We'll be seeing that in detail as the class goes by. So this disrupts the nuclear translocation of the SMAD4 and results in repression of transcription activation. So remember that CGMP action reduces or decreases transcriptional activation. So TGF has a role in hypertrophic and fibrotic remodeling of the heart. It regulates cardiomyocyte growth, fibroblast activation, and extracellular matrix deposition. So the SMAD proteins are signal transducers and transcription modulators that mediate multiple signaling pathways which are involved in cell growth, apoptosis, morphogenesis, and development and immune responses. So the atrial natriuretic peptide, you can see it in the box over here. It acts on guanylate cyclase, converts GTP to cyclic GMP. So the cyclic GMP activates the SMAD PKG, protein kinase G, which converts SMAD3 to the phosphorylated form. So this is the basics of the reaction, and further details will be dealt with later. So finally, you have the role of phosphorylation of tyrosine kinase. So we'll just see an example, which is insulin action. So insulin is a tyrosine kinase protein or hormone, and it's called a tyrosine kinase hormone because it has a tyrosine molecules in the insulin receptor. So this is the inactive form of the receptor, and when insulin binds to the receptor, which has the alpha and the beta subunits, the beta subunit, the alpha subunit undergoes a conformational change which is related to the beta subunit, and the beta subunit has autophosphorylating tyrosine molecules. It's called autophosphorylating because the tyrosine molecules have a tendency to undergo phosphorylation automatically without any external enzyme or help. So there is autophosphorylation of tyrosine, and IRS is nothing but insulin receptor substrates, which also has a tyrosine, which gets phosphorylated. So the receptor tyrosine kinases further phosphorylates other proteins, example IRS, and this leads to the activation of multiple signaling pathways. As an effect, it causes the biological effects of insulin. So remember that tyrosine kinase hormones or mechanism of action of tyrosine kinase hormones is based on the autophosphorylation capacity of tyrosine molecules in the receptor.